All right, guys, so, oh no, I don't want to do that. Here's that. Okay, so we're talking about DNA. Couple things I want to start with as a review, which I assume you're going to know because we have learned this already. Also, just a quick reminder, if you don't remember this stuff, your midterm is in 2.5 weeks and this stuff is on your midterm, okay? So first thing, DNA is what type of macromolecule? It's in the name. There we go, nucleic acid. That's what the Na stands for. Does anyone remember the other nucleic acid? RNA, ribonucleic acid, yeah. Okay, what are nucleic acids monomers? Nucleotides, good. There are 3 parts of a nucleotide. Do we remember the parts of a nucleotide? Raise your hand if you think you remember one. Nick?
Wait, no, wait. Let's think of something else.
Okay, Smith. A phosphate group is 1. A sugar, and then Justin? The what? That's a ribose sugar, yeah. And then, nitrogenous base. Good. Okay, good job. This is great. Okay, if you don't remember that, try. Okay. Okay, so now we're going to talk about the 4 nitrogenous bases. So DNA is made up of 4 nitrogenous bases in its nucleotides. They are adenine, thymine, guanine, and cytosine. It's okay if you just remember the first letters, A, T, G, and C. Okay. A's will always go with T's. C's will always go with G's. They are called complementary base pairs. Basically, they fit together like puzzle pieces and they match up. Okay, so they complement one another like peanut butter and jelly. Okay, except A's and T's, C's and G's. The Amoeba sisters in that video we're not watching, they say apples in the trees, cars in the garage to like help you remember A's and T's, C's and G's. Yeah, so if it helps you, great. All right, so in your notes on page 38, I would like you guys to highlight that first bullet point. The one that talks about nucleotides being the basis of nucleic acids. And then I would like you to highlight the 2 sentences that are in bold there where it says DNA's nucleotides are adenine, guanine, cytosine, and thymine. And then we have RNA. RNA has 1 different nitrogenous base than DNA does. So adenine, guanine, and cytosine are the same, but instead of T's, they have U's, okay? So U is for uracil, and those are specific to RNA, okay? So there's 2. Yeah, you have a question? Are you? I guess we have a couple pages, right? Okay. Hold on. Yep. Are we your next? Hello. I'm gonna go fast. Oh my God. 5 and 5, 9, 6, 5. 5, 5, 9, 6, 0, 5, 5. Moms. Okay, so I forgot. You're good. You can keep that. It doesn't matter. Just do that one. Okay, so 2 main differences between DNA and RNA you guys have to know. DNAs have T's, RNA have U's, okay? The other thing, DNA is made up of 2 strands. Anyone want to guess how many RNA are made of? 1, okay? That's the main difference between the 2. All right, now on your notes, before we go forward, does anyone know what the name of this like twisted ladder shape is? Double helix, perfect, okay. So you're going to highlight that bullet point that says DNA is a double helix, and then the 2 bullet points after that, all the way to the Watson and Crick. Okay. So all living organisms are going to have some form of genetic material, DNA, and the 2 men that are credited with all the work for discovering DNA, even though they stole it from a woman named Rosalind Franklin, are Watson and Crick. They're the one whose names are in all the textbooks, okay? Yeah. Much later, they credited her. Yeah, it was after people started being like, what the heck, okay? What? Yeah, people found her work and were like, this is predated to what they found, and they were in like the same lab or something like that and all that. Okay, so we're there with that. Do me a favor, real quick, close your eyes. Everyone close your eyes. Okay, tell me which base pairs with adenine? Thymine. And which one goes with guanine? Good, you guys are geniuses. Open your eyes. Okay, just didn't want to use on the board, all this stuff. I would like you up in the top where you guys have your little pictures that I drew that are almost like pointless. Please write for DNA and then for RNA. It's really, really, really important that you never, ever, ever use a U in DNA because it's wrong. And in RNA, you never, ever, ever use a T, okay? So they just basically exchange one. Yeah, I did the arrow both ways because it goes back and forth, yeah. What'd you say? Yeah, it is guac. And then for I don't know. Okay, so today we're only going to talk about DNA replication. So after you guys write the RNA stuff, we're going to just forget it for now. We'll go back over it tomorrow. Okay, DNA replication. Can anyone remind me why DNA has to be replicated in cells? Does anyone remember? Not quite. Dominic? Exactly. So when we go through cellular reproduction, mitosis, meiosis, once we do that splitting process, those divisions, we want the correct amount of DNA. For meiosis, we have to split twice, right? And we want to get half the DNA for, I said meiosis, right? Okay, good. And then for mitosis, we want to get exactly identical daughter cells, right? And so we do this process to make sure that we get that, okay? What stage of the cell cycle and meiosis does DNA replication occur in? Connor? Interphase, right? The first step for both. Remember that? Very important. Okay, so we call DNA replication semi-conservative because each DNA molecule is going to be made up of 2 strands. After we go through this process, 1/2 of each of the original strands will be in the new DNA molecule. Everyone cool with that? Yeah, it saves half. It conserves half, basically, is kind of the idea. All right. In your notes, under replication of DNA, please highlight the first 2 bullet points. There are 3 steps to this process: unwinding, base pairing, and joining. Okay. So there are 2 enzymes you guys have to know. The first one is DNA helicase. Helicase is going to unwind the DNA by breaking the hydrogen bonds that are linked up between the nucleotides. Who remembers anything about hydrogen bonds? What do you remember? They're weak bonds because they're what? Does anyone remember why they're weak? Good. It's just an attraction between positive and negative, right? So they're just like standing near each other, but it's like an attractive force, okay? So that means we can easily break them with an enzyme, which is what DNA helicase does. So this really corny pickup line you could say to somebody someday when you're much older: can I be the helicase to unzip your genes, right? Okay, that's what we're doing here. Okay, we're unzipping our genes. Okay, anyways, you're not gonna forget.
You were so close.
All right, and you know, do me a favor, highlight under unwinding the first 2 bullet points. So we're gonna unwind that DNA, break those hydrogen bonds using helicase, unzipping our genes. Okay, all right, now. Oh, no. Yeah, it breaks, it does, yes, pretty much. It goes between the two strands and breaks up the bonds between them. Yes. All right, so the next step is going to be base pairing. Base pairing is where the second enzyme you need to know, DNA polymerase, comes in and is going to one by one add in the nucleotides, okay, that are complementary to the original strand. So in the video, remember how they had a leading strand and it went just boom, 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 added them all the way through. And then there was the other strand where it added it in Okazaki fragments. Do you guys remember that in the video we just watched? So that happens, that's real, but for us and for me and my hand, we're just gonna go right through, okay? Is everyone cool with that? Okazaki fragments are real, that does happen on the lagging strand, but we're just gonna pretend like we can do it really easy all the way through. Okay? Yeah, pretty much, yes. Okay, so we're gonna start here at the top. You guys are gonna tell me what nucleotide I should try to this base with pair with this, and we're gonna add them in, okay? So we're gonna start with A. What goes with A? Keep going.
G, A, G, G, C, A, C, 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 G, 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 A, C, C, G, G, A.
Okay, look at it. Which strand is strand DNA? Woo! Okay, now that being said, what do you notice about our red newly formed strand of DNA and maybe something else down here? They're identical, which is what we are trying to do, right? We're trying to make two copies of the exact same thing. Did we do that here? Yes, we did. So on the lagging strand, we would do it in Okazaki fragments, but we're going to do the same thing. So what will our new strand look identical to? That top one. Okay, so I'll just do it real fast just because. Sorry, what'd you say? Correct. This will continue through the entire DNA strand. Helicase will continue to break the hydrogen bonds. DNA polymerase will continue to come through and add in the nucleotides until we end and get two brand new but identical DNA molecules. Everyone cool with that?
Yes.
How much of the DNA got conserved from the original in each of the new molecules? Half. All good? All right. Under base pairing, highlight the first bullet point. Here's my really awesome drawings. Thank you. Super talented. All right, and for the last one, do we understand what the word joining means?
Yes.
To join things, okay? So ligase is literally just gonna come through, it's gonna act like glue and it's gonna push together. So like, let's say there's something, I'm positive, there's a negative charge over there. We're kind of far apart right now. Ligase is gonna come through and just put us closer together. So our hydrogen bonds reform or reattracted to each other, okay? Yeah. Like the letter order, I just made that up. It can be in any order that your genes have, basically. I just want you to highlight the word joining, like the title of that section, as long as we understand what joining means, okay? And that is DNA replication. All right, and then by the end of it, as they get joined, the hydrogen bonds will then start to create that double helix shape again in both of the molecules. Joining, yes, exactly. There's a slightly better picture, right? Okay, close your eyes again real quick. Which DNA is responsible for unwinding the DNA? Which one? Helicase. And which one's responsible for adding in the nucleotides? DNA polymerase. Be careful because there is also RNA polymerase. All right, you can open your eyes. And we all understand 50% of the DNA is from the original thing. Fabulous. Okay, we're done with your notes. So you guys are all good with that. On this worksheet, I attempted to make it easier for you than I have for kids in your class because they get a little confused. At the bottom here, where it says writing a full strand, this whole bottom section, part A is your original DNA 2 strands, okay? you have to write your complementary base pairs for that, what they gave you. All right. Then B is basically you just opening up the two strands. I tried to draw your DNA picture, kind of similar to the one on the board. Those are opening up, and then you're just going to add in the nucleotides. They should be identical to each other. Okay, you should have the same answer for this as you do for the next two and the next two blanks. Okay, is everyone kind of clear on that? If you're not, you can ask. On the back side, just in general, think of a molecule as having 100% of the nucleotides, right? Because they're all made of nucleotides. If 15% of my molecule, of my DNA molecule is made up of A's, what percentage are T's? 15%, because A's and T's go together. That's 30%, which means how much total are my percentage for G's and C's? 70. So then that means that there will be 35% G's, 35% C's to make that 70. Is everyone clear on that? That's just a little bit of math you have to do on the back. Okay? Do tomorrow, or if you finish today, you can show me today. Okay? Good luck.